messieurs, je vais en effet vous parler de l'homme de l'an 2000.
Ladies and gentlemen, I'm here to talk to you about 21st century man. Actually, it's not exactly 21st century man I'm going to talk about, because humans won't have changed by the year 2000. Humans are a very stable species, a species that hasn't changed in 50,000 years. And it's quite probable we will never change again. Although humans won't undergo a major biological transformation, scientific progress will significantly modify the human condition, human mores, and in particular, human reproductive behavior. Scientific progress has a tendency to technologize, if you will, or biologicize the human species, gradually transforming it from a natural species, or Homo naturalis, into an artificial one, or Homo biologicus. One of the changes that will occur with absolute certainty is that by the year 2000, humans will have a much longer lifespan.
In 1958, the French scholar Jean Rostand predicted the advent of a bionic man with enhanced capacities that could be repaired like a machine and would never grow old. With the discovery of the genetic code came the extraordinary possibility of one day mastering our own biology. Today, biologists are liberating themselves from the laws of evolution. They manipulate living matter, penetrate inside cells, intervene in their internal programming, activate, inhibit, correct, and modify genes. Genetics is the key to understanding human biology, and the race to unlock its secrets is accelerating. A team of researchers at the Massachusetts Institute of Technology discovered a gene that prolongs life in all living organisms, from the most primitive to the most complex. This gene, activated in the laboratory by subjecting mice to a restricted diet, could very well be the gene that determines longevity.
So this is about 2.5 grams of food. It's about how much each mouse will get every day. They're usually very hungry, and as you can imagine, they would lose weight, and they are lean. They have a low blood glucose, blood insulin, triglyceride, and cholesterol. So these are all very beneficial to their health.
Calorie restriction is a dietary regimen where you reduce the number of calories that the animal eats to roughly 60% of normal, while maintaining the same number of vitamins and minerals. So the animal isn't actually malnourished, it's just getting less calories. And that extends the lifespan of the animal quite dramatically, and also provides resistance to a wide number of different diseases, including some kinds of cancers, diabetes, obesity, and some neurodegenerative diseases.
This is a fascinating finding that we've known about for a long time. In fact, I think it's by far the most robust way to make a mouse or a rat live longer. But what's been lacking all these years is an understanding of how it works. How is it that diet regulates aging and lifespan? And so this hypothesis that maybe SIR2 and its relatives could be the agents that delivered the benefits of a low-calorie diet was, I think, a very exciting prospect. In fact, it took many years, really 8 years, before we settled on the notion that SIRT1 gene was an anti-aging gene. That is to say, if we increased the activity of this gene, the cells lived longer, and if we decreased the activity, they lived shorter. And what's quite striking is that if you knock out the gene altogether, the mice are quite sick. So this is a gene that in mammals, it's important that it be present. But what's interesting is if you raise the amount of expression of this gene, that the mice adopt many characteristics of calorie-restricted mice. So this, to my mind, fortifies the hypothesis that the SIR2 gene, and in particular the mammalian SIRT1 gene, will mediate at least some of the effects of calorie restriction. And so, for example, the mice with superactive SIRT1 are very lean. They have the blood chemistry characteristic of calorie-restricted rodents.
So what kind of, even like marriage, it's not very common for people to get married to a drug. You know, so it's more common for
To continue that path, what one would like to do is to develop a menu of drugs to sirtuins, SIRT1 obviously, but maybe to the other mammalian sirtuins, SIRT2 through 7, and to test these drugs first in animals against various diseases, and then to bring them to the marketplace by testing in humans for benefit against particular diseases. So I think this is the research track that I see playing out in this area of aging and aging-related diseases, is to now develop what we know about important genes and their products, like sirtuins, to develop drugs that will give benefit for diseases of aging.
Information technology is doubling its power every year. Example, when I was at MIT, a computer that took up actually more space than this room, this conference hall that you're in, cost $11 million, was shared by all thousands of us, and it was really the most advanced computer of any university in the United States. This computer in your cell phone today is 1,000 times more powerful. And we see the same thing in biological sciences and technologies. And we're now really at that early stage of understanding biology. Yes, we've collected the genome. We're in the early stage of really understanding it, reverse engineering how the genome works, how the brain works, how these biological processes underlying disease work. But all of the tools are progressing exponentially. We're doubling the power every year of the ability to simulate, model these processes, reprogram them. And so these will be 1 million times more powerful 20 years from now, and that really will be very transformative.
Fueled by the conviction of a few visionaries like American physicist Ray Kurzweil, more and more scientists are being lured by the promise of a golden era of biotechnology. Research is progressing at an increasingly rapid pace as scientists in every part of the world pursue new leads and innovations. We are witnessing the emergence of a revolutionary field in medicine, regenerative medicine, that may one day be able to reverse the aging process. Just a few years ago, biologists succeeded in extracting a cell with extraordinary properties from an early-stage human embryo, the embryonic stem cell. Embryonic stem cells have the unique ability to renew themselves and replicate indefinitely without degenerating. Not only can they live forever, they can also differentiate or transform into any type of cell in the body. Depending on the information they receive, they can become muscle, skin, blood, bone, brain cells, or heart tissue. They provide an unlimited reserve of fresh cells that could repair and rejuvenate damaged or aging tissue. Another laboratory at MIT is engineering human tissue by growing stem cells on synthetic scaffolds that biodegrade within the body. The day is not far when they will be able to engineer entire kidneys, livers, hearts, spinal cords, and blood vessels.
There's multiple phases in growing a tissue or organ. One phase is growing the cells, is getting the right type of cells, which could be stem cells, and convert them into the right cell type, or it could just be isolating the particular type of cells you need, like cartilage cells, bone cells, and so forth. Another area that's also very important is the materials. What's the right materials for the cell to grow on? What's the right way to manufacture that material to give it the right shape and so forth? We then combine the two. We add the cells to the plastic scaffold, the material scaffold, and then grow it in what we call a bioreactor. And in that stage, it needs the right nutrients and so forth. And then we take the tissue that we create in that bioreactor and actually put it into the animal or the patient. Well, we've worked on about over 20 different tissues and organs over the years. We've worked on spinal cord, we've worked on intestine, bone, are doing some very exciting work on trying to tissue engineer new hearts.
So here is an example of some scaffolds that we have made for stem cell differentiation, in particular for bone regeneration. And these scaffolds are biodegradable in nature, which means that they can degrade so that they will be absorbed by the body. And most importantly, the shape can be molded to whatever thing you want them to become. For example, we can make like an ear shape, we can make a nose shape, we can make a chin shape from the same material. Trying to make a piece of tissue is very similar to trying to build a house. So cells are like the bricks when we're trying to make a piece of tissue. And also the scaffolds provide structural support, initial structural support.
And one technology we have is microfabrication technology at our disposal. And what we're able to do is we're able to use the same technology used for microchips. So this is developing structures on a very, very small scale, the micron scale. And we're able to use those technologies and interface them with biomaterials. And here I have a silicon mold that mimics the right fluids of the blood vessels that are in your body and mine. And so from this mold, we are going to cast polymer and novel biomaterials onto this mold for eventual use as a mimic of blood vessels found in your body. And I have one of those polymers right here. This polymer is actually very flexible, just like some of the tendons and ligaments in your body are flexible. Another important aspect of those biomaterials is that they are biodegradable. And what this means is that we can implant this material within your body, and over time, those materials will degrade and eventually disappear within your body. And here what I've zoomed in upon is a microfluidic network, which simulates an array of a capillary bed that you'd find within your body. And if we look closer, what we see is micron scale channels that, just like your capillaries, span out and perfuse and feed the cells around your organs.
Here we're looking at a specimen of tissue-engineered heart muscle that we've fabricated by combining neonatal rat heart cells with a novel scaffold material. And the structure here that we're seeing, we're terming an accordion-shaped pore structure. This accordion-shaped pore structure was designed to help mimic the mechanical properties and the structure of the actual heart muscle tissue. And we can see that within this pore structure, the pore is about 400 microns in length and about 200 microns at its widest point. The heart muscle cells filling throughout the pore structure and contracting synchronously.
Right now, we're starting very small with a little cardiac patch to be put onto an area that might have been damaged by a heart attack, or a vascular graft that might be put on an aorta or another part of the heart. There are other people that are working on tissue-engineered valves, and so you can envision eventually maybe these approaches could be combined into a full organ repair, but that would be further in the future.
The real goal, the eventual goal, is to be able to fabricate tissues and organs for any patient that might need it. For example, if you need a new liver, if you need a new heart, if you need a new lung, you can simply create a construct which can be produced by essentially a factory, and these organs can be pulled off the shelf and implanted in you, and so you can get your new liver, heart, or lung.
As science comes closer to mastering the full potential of stem cells, the demiurgic dream of creating living body parts to replace damaged ones and reverse aging is on its way to becoming reality. Aubrey de Grey, an English geneticist at the University of Cambridge, has developed a radical theory about extending the human lifespan. He's convinced aging is like any other curable disease, and that death is not inevitable. A controversial figure, he is one of only a handful of scientists to postulate a hypothesis on immortality.
Something that I think is true is that the first person to live to the age of 1,000 has very probably already been born. I would say there is an 80% chance of that. I would say that there is a 50% chance that the person who will first reach the age of 1,000 is already 40 or 50 years old. We already know that aging can, in principle, be repaired, simply because we know that the human body is a machine. It's a really, really complicated machine, and we don't have the plans the way we have for man-made machines, but it's still a machine, which means that its functional lifespan can be increased indefinitely by sufficiently thorough repair and maintenance. So in other words, there is nothing more to discover to prove that aging can, in principle, be repaired. What we do need to discover is the details of how to implement these various repair and maintenance strategies so that we will actually have the technology.
We understand how a house works because we created that methodology. We understand all the methods underlying a house, so we can repair them. We don't yet have that knowledge, that set of tools with our bodies, but we're getting there very quickly, and we will get there exponentially. And in my mind, actually, I think Aubrey's projections, although they're considered radically optimistic in some quarters, are, I think, a bit conservative, perhaps by design. In my mind, within 15 years, we will reach that escape velocity where each year we're going to be adding more than a year, not just to infant life expectancy, but to your remaining life expectancy. Now, that's not a guarantee of immortality, but that is an escape velocity. So as you go forward in time, the amount of time you have left actually increases rather than decreases, and that'll be a very different experience.
Utopian of projections, humankind will invent the means to enhance its capacities by directly interfacing the body's biological systems with the most recent technology. Soon, reprogramming our own biochemistry will be as easy as reprogramming a computer. The technology of the infinitely small is already producing the first prototypes for use in nanomedicine. Machines the size of molecules that will operate directly inside the human body and deliver drug treatments into cells. For nanobiologists, these molecule machines will revolutionize the future of medicine. Soon, not a single disease will escape their field of action.
Mankind has historically built everything from simplicity. We've always built complexity from simplicity. During the agricultural and mechanical ages, we built everything from 6 simple machines: levers, pulleys, screws, and everything that we built a whole period of time in our history has been with these 6 simple machines. During the electronic age, we built everything with 5 simple machines: resistor, a capacitor, an inductor. And the result of this is that it really limited the complexity of the devices that we could build. Nature, on the other hand, has a palette of tens of thousands of simple machines. And what I wanted to do was I want to have access to this incredible palette of material from which to build devices and machines that have the complexity that's comparable to what you find in living systems. So as a result of that, we have embarked on first trying to figure out how in the world do we harness these individual molecules and assemble them to do things that we want to. And what you're looking at here is one of the first devices that we made. This device is 125 microns from head to tail, and it's fully self-assembled, and it walks along using the energy of these cells come from the hearts of rat pups, cardiac stem cells, to propel them. Once we're able to do this, the concept then became, can I take this machinery and start making it into devices, into rods and levers that we could ultimately insert in the body? So working with this type of material is very exciting, but we really wanted to be able to get deeper down into the physiology of living organisms. We wanted to be able to make devices that we could insert at the most fundamental level of living organisms. So our work began with a molecule called ATP synthase. This is the powerhouse of the cell. And what happens is there's a rod that spins around. As this rod spins around, it moves some cylinders that move back and forth. And this flexure causes the molecules of ADP and phosphate to generate ATP. What we did was we took this portion, the engine portion of it, we disassociated it from the membrane portion, the turbine. We took the turbine, we flipped it over, and so now we have an engine with a shaft coming out. And then what we did was we stuck a nanorod on top of the shaft. So as the pistons were moving and the shaft was spinning, the rod, which made it look like a propeller. That is the original nanocopter. So it's really quite quaint now. But when we're doing it, we had to solve a bunch of problems. We had to learn how to take these molecules and disassociate them, how to develop chemistries to make them attach them exactly where we want to, how to put those molecules where we want to. And they had to be in the right orientation. They couldn't be laying down. They had to be exactly where they had to be. Again, it's nanotechnology.
Okay, how much? Well, let's do something like a 1:100 ratio.
1:100 的一个 ratio. Okay. Suppose that is 10 microliter.
Oh no, I mean 1:100 milligrams.
Okay.
The concentration is on there, so it's possible to calculate it.
Okay, I will calculate that. Okay, thanks.
Actually, I'm working on a project using a molecule called 529. It's a RNA motor. It's the packing motor that's used in viruses to pack the DNA inside of the capsid of the virus particle. And we're using this now to engineer a device that would pack the DNA inside an artificial device that we built and then inject it into a cell for a mechanism of gene therapy. The viruses have a container on top. And at the bottom of that container is this motor. And this motor really, what it does is it spins around, and as it spins around, it takes the coils of DNA and it shoves it up inside of this container. So it spins around, the DNA goes up and gets packed up inside there very, very tightly. And then what happens is, after the DNA is replicated and is placed inside there, when the DNA is packed so tightly, it's under incredible pressure. And what happens is, when this virus particle gets to a cell it wants to infect, it gets on the cell, and a door opens up, and the DNA just shoots right inside there. Because on this high pressure, it shoots right inside there, and that's how the cell gets infected.
It's just a very strong motor. And it's, in fact, actually one of the strongest motors that people have found in nature. And so you can imagine that a very strong motor might be useful for moving things that are much larger than itself. So we're trying to take it out of its natural architecture and put it into something new.
Working with hybrid systems, living, non-living, unified systems, provides you the opportunity of interfacing devices that you engineer seamlessly with living systems. What I'm looking at is trying to engineer materials and devices so that when I place them inside a person, that they integrate with the life cycle of that person, they integrate with the energy flow, with the physiology of that person, so that they become part of that person. Instead of being this ugly, at least aesthetically, ugly box that you stick on somebody, what you really are doing is you're engineering a component that provides a function that when it's placed inside that person, you can't differentiate where this component begins or ends and where the living person ends. The interface is so seamless, they meld together.
But if we go out to 2010, I think this technology is going to disappear. We're not going to see it. Devices which are already about this size are going to become so small they'll be embedded in our clothing. Images will be written directly to our retina. We'll create full immersion virtual reality environments from our lenses. And we're going, in the next decade is really when these biological technologies will come into fruition. We really will have the means of simulating in great detail the biological processes that lead up to the different diseases that kill us. And we're gaining new insights as to how the brain works, giving us more insight into ourselves. And this will help both with the project of achieving engineered negligible senescence on the one hand, but also this other project to expand our horizons by creating more intelligent machines, which are really an extension of ourselves. And literally, we're going to, in my mind, put them inside our bodies and brains to literally expand our capabilities.
Recent progress in the field of neuroscience has opened up exciting but sometimes disturbing prospects. Neuroimaging, magnetic stimulation, and neural prostheses provide new possibilities for exploring and repairing the brain. For over a decade, Neurobiologists at the University of Southern California have been developing a neural prosthesis, a synthetic memory substitute. In as little as 15 years, this device could reverse damage caused by neurodegenerative diseases, such as Parkinson's or Alzheimer's, and eventually eradicate senility. Could these new prosthetic devices be the final phase in achieving a healthy mind inside an immortal body?
Imagine that your goal is to understand how the brain creates memories, right? And we have a lot of theories about how the brain constructs memories and how it recalls memories, and they're very cognitive, they're very high level, they're very conceptual. How do you take a conceptual understanding of the brain? And it's not that it's wrong, it's probably quite correct, but how do you take a conceptual understanding of the brain, and how do you reduce that to a microchip? How do you do that? You know, it's not an easy thing to do, to move these concepts into circuits, into mathematical equations. And I think that, and some people think that our attempt to build neural prostheses for things like memory or for cognition, they think we're crazy. And I think the problem is that people keep thinking about cognition, language, memory, and they think of it in terms of these concepts. Well, why not think of it in terms of signal processing? Because in the end, that's what the brain does. That's what neurons do. They receive electrical signals, they change those electrical signals, and then they have an output of more electrical signals. So let's give a neuron all possible inputs, which would be a series of these random intervals. We stimulate the neuron with it, and then we look at the electrical output of the neuron and ask, what is its output to all of these random inputs? And so we generate a very general model, a model that shows us how real nerve cells would respond to any conceivable pattern. You can give me a dot, 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 any pattern you want, give it to me, and I can program a computer, and we can predict what the output of the neuron will be, and it'll be exactly right. In reality, we probably don't need to know much more than that to be able to replace a part of the brain.
Here's a hippocampal slice. It has 3 regions. The signal passes from here to C3 and propagate from C3 to C1. So if we make a cut between those 2 regions, like what we did here, and we send stimulation to dentate, there'll still be activity in dentate region, but you won't see any response in C3 and C1 because it is damaged. And we did this to mimic what happened in patient where C3 region was damaged. And finally, we built this FPGA chip to replace the damaged brain region. So what the chip does is mathematically predict what the stimulation should be to C1 region. So if you only look at the output signal in this replacement experiment, you won't see any difference between this and what you observed in the intact slice. And in other words, we functionally replace the damaged C3 region with this FPGA chip.
Brain chip doesn't really repair the damaged region, it takes the place of the damaged region. It's a substitute. And that's what we're trying to do. We're creating a substitute.
And what we have to accomplish is not only getting the mathematical model correct and the microchip correct, but then we have to be successful in getting the microchip to communicate with the brain, and that's not easy.
Ça, c'est possible que dans l'avenir, on puisse
In the future, it's possible that a neural prosthesis, when inserted in the right place, could reactivate certain memories that have been forgotten.
Réactivation de certaines mémoires qu'on a pu avoir oubliées. Par contre, si il y a certaines
However,
mémoires
if a person has completely eliminated certain memories,
alors est-ce que chez
will
ces
we be
gens-là,
able to recreate
on
them?
va pouvoir
Will we
recréer
be able to recreate
information?
that information?
Je ne sais pas.
That I don't know.
Ça, c'est toujours aussi la même chose. Est-ce qu'on pourra, dans le futur, faire, par exemple...
And the same goes for artificial memories. In the future,
faire un
will we
transfert
be
de
able
mémoire d'une
to,
personne
for
à l'autre?
example,
C'est-à-dire que si une
transfer
personne
memories
a
from
sa
one
mémoire,
person to
est-ce
another?
qu'on peut
There are
les
movies on that
transmettre
subject.
directement, donc,
Will it be possible to transfer someone's memories directly via a system of
circuits intégrés,
integrated
communication de
circuits
cerveau à
from one brain to another,
cerveau, circuit intégré,
or from one brain to an integrated circuit to another brain, and recreate artificial memories? That's a really difficult question.
question difficile.
Reducing human intelligence to a set of equations seems nothing short of impossible. And yet, a team of neuroscientists from the Swiss Federal Institute of Technology is creating a virtual brain, a perfect replica of a rat brain, on one of the most powerful supercomputers in the world. In 10 years' time, when the project is completed, the Blue Brain program should be able to simulate and visualize the electrochemical functions of the brain it's modeled after. It's possible that from the interaction between the billions of neural connections, a glimmer of intelligence could arise. Scientists hope this brain simulator will help them discover the secret to human consciousness.
So what you're seeing here is a fly-through of the neocortical column after we've built it. It's about 10,000 cells. We're only seeing about 10% of them. And the important aspect of this is that you can see that 90% of the brain is branches. And this model, the Blue Brain project, allows us to actually understand and explore the function of these branches. And there is no other way to explore all of these branches working together other than putting them together, building the circuit together. In the end, it is this module, this processor, that will allow us to understand human intelligence. So even though we study this at the level of a low mammal, such as a rodent, it's the first step to be able to go up and understand the core circuit properties in the human.
We actually have tremendous facilities in the laboratory for recording from individual neurons, injecting dye into the neurons, and using the biology, measurements of the biology to reconstruct the full three-dimensional structure of the neurons. the full electrical behavior of the neuron. So we have a suite of stimuli that we apply to characterize how a neuron responds to input. And then we combine these to build a single model of a neuron. And then we place these neurons within the volume of a neocortical column such that they correspond to the biological observations of where these neurons occur in the different layers of the cortex.
We do in vitro preparations. So that means essentially we take slices of brain which need to be prepared. So essentially you take out the brain and then shock freeze it, you cut the slice of the brain, and then essentially let this slice rest. You mount it on an object carrier and then put this under your microscope. So essentially once you put the slice under your microscope, you mount the electrode with a glass pipette on top, very thin pipette so that you can penetrate the membrane of a cell, and then mount an electrical connection to the inside of the cell.
I'm positioning the tip of the pipette in the direction of the cell. I'm moving the pipette towards the cell. So here, we're approaching the cell. We can't see it yet. Now it's touching the surface of the membrane. We have contact between the pipette and the membrane. We're ready to begin recording cell activity.
So this gives us a tool in which we can actually replicate experiments that have been performed in the laboratory and compare the results in the model directly with the reality, and then ultimately make predictions and understand in a more deep way, because we can perform experiments that are simply impossible in the real brain circuitry.
In 10 years, you can imagine that a researcher would put on goggles, enter into Blue Brain, he would enter into a virtual laboratory, and in that virtual laboratory, he'll be able to run all kinds of experiments. We will couple the brain with, for example, a virtual reality entity, an avatar, that will have a blue brain on it, sitting on a supercomputer. You can also connect it not only to a virtual avatar, but actually to a robot. So you could have a physical robot that would be interacting with the world through cameras and recorders, and this information would be injected into the brain, and then it would send commands to drive the robot. So if we build it wrong, this robot is just going to crash, fall, not be able to do anything meaningful, not be able to communicate, and not do anything intelligent at all. Whereas if we build it correctly, we should be able to see it learn like a baby, acquire knowledge, and start to move correctly, respond to stimuli, and communicate. If we finally build the human brain correctly, it should speak a language. You should be able to teach it a language, and it may be able to teach many different languages. So, and then as you teach it a language, you'll be able to see where does the language go, how is it embedded, is it distributed, is it very localized. So you'll be able to ask some very important questions about language generation, and then during the process of language, you'd be able to track every little step and understand this tape recorder that just comes flowing out of our mouths when we speak. How are they coming? Where do they originate from? Where is the mind? Where is the beginning? When I say I want, what is it? What drove that sentence? So with this kind of technology, we should be able to dig down to the very origin of thought.
Ethical barriers are gradually breaking down and won't stand in the way of scientific progress much longer. But could the unbridled quest to master our own biology be a race with no end? The ideal of perfect health an illusion? Perhaps one day we will succeed in warding off death, engineering new genes, new organs, new memories. But somewhere along the way lies the risk of altering the very core of our identity.